Yes. Thanks, Vince. We've known for a very long time, and Michael kind of touched on this, that this does not look like my normal slide. So neuronal dynamics are critical for behavior. And I'll give some really seminal examples in the domains of learning, memory, and working memory. So in terms of learning, dopaminergic neurons will naturally fire or increase their firing when a rewarding stimulus is experienced. If that stimulus becomes predictable, they dynamically shift the timing of this increase in firing the neurons do to begin to respond when that rewarding stimulus is anticipated. And they no longer respond when the reward is received. Now, it gets very nuanced. So if a reward is predicted and then it does not occur, there's a transient dip in neuronal firing. So what this is demonstrating is these dopaminergic neurons dynamically shift their responding and their patterns of responding based on the environment, and this is how we learn. In terms of memory, this is a figure showing long-term potentiation. So each of these arrows here are time points when hippocampal neurons are stimulated. And what you see is, of course, when a neuron is stimulated, it fires more. But this, long hours after stimulation ends, these neurons still remain elevated in their degree of firing. And this is an example with tetanic stimulation, so artificial stimulation. When external stimuli are encountered in the environment, the exact same process of long-term potentiation occurs, and this is how we encode and remember new information. And in terms of working memory, so during working memory, one must maintain a stimulus, an item in memory across a brief delay period. Dorsolateral prefrontal cortical neurons are active during the delay, but not indiscriminately. That's showing, yeah, whatever. Not indiscriminately. So what this is showing, this is a tuning curve. So in a spatial working memory task, and this is the same for other domains, neurons will fire selectively to stimuli in a specific location. And they'll fire either less or not at all if stimuli are in other locations. And this is directly relevant to maintaining that item in memory. So for example, here we're seeing an item that has to be remembered that's in this location. If there is this increased inactivity in this neuron and other neurons that fire with this pattern, the stimulus is successfully remembered. Same stimulus, same tasks, same exact external environment. If those DLPFC neurons do not fire during the delay, that stimulus is not remembered. And so this is a great example of how these dynamic changes in the patterns of neuronal firing directly impact whether or not our cognition is successful. So taken together, can I say something? This is missing a lot of my slides, and now it's fine because all the figures are there. I'm worried some of the figures are going to be missed. Can it be made smaller? Sorry, I'm missing the tops and bottoms, yeah. And I'm worried something critical will be missed at some point. But so those are examples that just demonstrate that these neuronal dynamics are really critical for cognition. And a point that I wanted to make with that is what the dynamics are shift based on the context. So sometimes they're time varying, which we've been talking about a lot today. So in learning, right, sometimes firing increases, sometimes it decreases within a small period of time. Sometimes it's time varying, and that's what's really critical. Other times, the dynamic shift is to make neuronal firing more stable and more sustained and more consistent across a period of time, like in the working memory example. And so these shifts toward greater time varyingness or greater stability are both really important for cognition. We can see, I'm sure it'll get most of the slides. Oh, what's happening? I'm not seeing the titles. So the next point I wanted to make is that we obviously cannot go around measuring neuronal dynamics in healthy humans. So as Michael talked about, there's been a lot of really great modeling work linking neuronal dynamics to functional MRI measurements. There's also been a lot of great empirical work that's been linking, attempting to link various levels of neuronal dynamics to functional connectivity as measured with fMRI. So these papers are some examples of relating LFPs in animals to fMRI signals, time-varying functional connectivity. Here's an example relating ECOG in humans to fMRI, and these last two are just examples of relating EEG to fMRI measurements in humans. So this is the general outline, and there are two main bodies of literature. What I'll do is I'll review some of the literature that relates time-varying functional connectivity as measured with fMRI to cognition. So what we're basically doing is we know these neuronal dynamics are really important for cognition. We also are developing these techniques, and Tim will talk a little bit about this, I'm sure, to get at reliable estimates of these neuronal dynamics, or like proxies for them using fMRI data. So I'll talk about some empirical literature as it relates to cognition. One, some literature investigating how different time-varying patterns of functional connectivity relate to individual differences in cognition. So this is looking across subjects, individual differences. And then I'll talk about some literature investigating how trial-by-trial changes in functional connectivity relate to changes in performance within an individual. So this is time-varying changes within an individual impacting, being related to cognitive changes within an individual. And then some conclusions. So in this first study that I'll talk about, this is one of the earlier studies by Elton and Gao, I think in 2015, that related time-varying functional connectivity to behavioral performance. They gave subjects a stimulus response compatibility task, easy and hard blocks. And this first figure that I'm showing is, what it's doing is they did a sliding window correlation that has been talked about before. And these matrices are measuring the variability of that functional connectivity strength across time. And so this is the first task block, the easy task block, the difficult task block. And the two task blocks were flanked by two resting state blocks. And what you see is the variability of the time-varying functional connectivity decreases during the task blocks. What they then did is they calculated graph theory metrics on each of the windows and measured also the variability of the whole brain network organization across time in these resting state and task blocks. And what they found, they looked at the whole brain metrics of global efficiency, of local efficiency, and of modularity. And they found that in all three, variability of these network organization metrics decreased during task. So network organization remained more stable during task. And in some cases, it became even more stable during the more difficult task. And when they related this decrease in functional connectivity variability to behavior, what they showed is that, so this is the easy task and the hard task, more so in the more difficult task when probably subjects have to concentrate more to engage in the task. This is looking at the decrease in variability, that's what the x-axis is. If variability decreased more from the resting state to the task state. So if functional connectivity became more stable and network organization became more stable, participants performed better on the task. So this is a great example of showing that these shifts from a resting state to a more constrained task state, how that shift occurred directly impacted performance. This is a study that Vince was involved in. This is measuring mindfulness, mindfulness in children and in adolescents. And what the group did is looked at time-varying functional connectivity between the salience network, default mode, and central executive network. And they calculated k-mean, or they used k-means clustering to develop brain states. And this is just depicting the 5 different states that they pulled out of this data. And if you measure the number of state transitions, so the number of times throughout the course of a resting state scan that functional connectivity organization transitioned from one of these states to another one of these states, not caring which state it is, the more state transitions there were throughout the course of the resting state scan, the greater the mindfulness was reported in these individuals, in these children and adolescents. And then what this is showing is something very consistent in the literature, is that those subjects who reported more mindfulness, more mindfulness reported less anxiety. And so the next step they took was a mediation analysis where they showed that the number of state transitions, so this time-varying functional connectivity, the greater the variability, that mediated this relationship, this negative relationship between mindfulness and anxiety. So what more can we learn from research like this? I'll talk now about, this is data, a study that's ongoing that data I collected with Mark Desposito at UC Berkeley. We did this was in healthy adults. They completed 2 resting state scans, one week apart from each other, and then immediately after the resting state scans, completed a battery of cognitive tasks. And I'm really interested with this study in looking at differences in state. So do differences from session to session in an individual in terms of time-varying functional connectivity relate to differences in behavior that we see from session to session in those individuals. And then I used k-means clustering and identified 3 connectivity states separately for each of the 2 sessions. And consistent with a lot of work, I think Vince mentioned this briefly before, doing this separately for each session, what you see is these states are very reliable across sessions. So individuals were fluctuating across these 3 states. These 3 states were very similar in session 1 and session 2. So what I'll show you now is how number of state transitions relates to performance on a Stroop task. And I'm particularly interested in the most difficult, the trials that require the most cognitive control, so these incongruent trials that I circled here. And I'm showing differences because, like I said, I was interested in change from session 1 to session 2. So to describe, I've lost the x-axis here, but the x-axis is change in number of state transitions. So if there were more state transitions in session 2 than in session 1, that would be over here. That would be positive values. Whereas if there were more state transitions in the first session as compared to the second session, that would be negative values. And on the y-axis, we have behavior. So I'm looking at reaction time measure. So lower is better, right? So if participants performed better on the task, if they were faster, if their response times were more stable, then these would be negative values on this plot. This means they were better during session 2, better during session 1. And what I found is that in participants who decreased their number of state transitions during session 2 as compared to session 1, they performed better on the task. So in other words, this more stable connectivity, less of this time-varying nature, may relate to a greater ability to sustain the attention and exert the cognitive control needed to complete this task. This is similar to that first example that I gave you. And then so where else can we go from here? So in terms of understanding the mechanisms of cognition, one of the directions we should go is understanding when is it adaptive to have more stable periods of functional connectivity. Like during the delay period with working memory, perhaps when information or attention must be sustained across time. But in other situations, like the mindfulness example, it seems that it's more adapted to have more dynamic periods of functional connectivity. And so perhaps during periods of cognitive flexibility, it's more adaptive to have these more dynamic or time-varying periods of functional connectivity. And in terms of clinical relevance, which is important to me in my research, one of the specific questions that comes up with this research is how do these brain state transitions facilitate decreases in anxiety specifically, and more generally, how can we develop treatments that target these functional connectivity dynamics once we understand what goes awry in certain disorders? How can we use that information to help treat patients? So now I'll talk about some literature looking within individuals how changes in functional connectivity patterns impact differences in performance on a trial-by-trial basis. So in this example, this was a task. This is by Sepida Sadagiani and Mark Desposito and colleagues. This was a task where subjects heard an auditory stimulus periodically and rarely throughout a scan. It was peri-threshold, meaning it was very hard to detect, and it was subject-specific, so subjects missed about 50% of them, heard about 50% of them. And then what they did was they took these periods just before the stimuli occurred and looked at connectivity patterns during those periods and clustered them into hits and misses to see if there were systematic differences in functional connectivity just before someone heard an auditory stimulus or someone did not hear that same stimulus. And they looked at functional connectivity between cognitive control, attention, default mode networks, and auditory network. And this matrix is showing the difference in connectivity strength before hits versus before misses. And these black cells are showing connections where the connectivity strength significantly increased or was significantly increased before hits, before misses. So this is just a global view of showing that there are systematic differences. They then computed modularity, and they have here on the left pre-hits modularity and then pre-miss modularity, these same time points. And what they found is that modularity was significantly decreased before trials on which subjects missed the auditory stimulus versus trials on which they heard they detected the auditory stimulus. And when they broke up this modularity to see which networks were driving it, it was the default mode network shown up here and the visual network shown down here. So when the default mode network, as well as the visual network, which is a sensory network that's not relevant to this task, when those two networks were more integrated into the whole brain network, subjects were more likely to miss the auditory stimuli. Oh, that looks better. Now I see my titles. So this next task by Aaron Kusi and Yves Valera, they did a self-paced finger tapping task. So they just had subjects tap their finger. They asked them to tap it regularly throughout a scan, but let them self-pace. And the idea here is that if someone is paying attention, the tapping will be very steady. If you get distracted for whatever reason, the tapping will become more variable. So this is just showing the variability of tapping across time, and you can see it fluctuates. And what they did here is they looked at which functional connectivity patterns that were related to higher variability in tapping rate, using this as a proxy for inattention. And what they found is that, so here they're depicting 3 different seed regions, and the anterior insula, so a region of the salience network, and then 2 regions, MPFC and PCC, of the default mode network. And what they found for both within default mode network and between salience and default mode network, the stronger the functional connectivity, the more variable the responding was. And so what more can we learn from this? This research makes me think of this default mode interference hypothesis. So both of the studies that I just talked about indicate a key role of the default mode network in being able to successfully attend to and perceive stimuli, which is consistent with this default mode network interference hypothesis, which states that intrusions of the default mode network into task-positive networks underlie attention lapses. So this is just depicting the default mode network in green, a task-positive network in yellow, and the idea is, right, this is familiar to all of us, during a resting state, they're anti-correlative. So if activity in one is higher, activity in the other is lower, and vice versa. And this figure is a really nice depiction of what they think is going on. So here we see during rest these very strong anti-correlations between default mode network and task-positive networks. And here they talk about it in terms of introspection, the default mode network being thought to be related more to introspective, internally focused experiences, and task-positive networks, which is thought to be more related to extrospection or attention to external task-relevant stimuli. And so what they're showing is at the initiation of a task, these anti-correlations don't disappear, but they get much smaller in magnitude as attention is directed to the task. And this red line here showing behavioral performance, which and what you'll notice is there's a relationship. The lower the magnitude of these anti-correlations, the better the task performance. And as they're implying here, time on task is related to decreased attention, but obviously it doesn't have to be linear. But what they're showing here is as behavioral performance wanes, as attention to the task wanes, and performance starts to become more variable, these anti-correlations get higher. And the key part being that if default mode activity passes this threshold, then it starts to interfere with task performance. And that's when you get these attention lapses. So this is really relevant to me. A lot of my research is focused on ADHD, and children with ADHD and adults with ADHD have a problem with inattention. So I'll talk about some research I'm working on right now with Stuart Mostovsky at Kennedy Krieger Institute in children with ADHD and typically developing children, matched for a whole host of variables. And we measured intrinsic brain organization during rest, and I measured both static connectivity, so functional connectivity across the entire resting state scan, and then I also looked at time-varying functional connectivity. And here what this is showing is I calculated modularity from those connectivity matrices, and what we found is that children with ADHD display higher modularity as compared to typically developing children. they also display a higher variability in that modularity across time during the resting state scan as compared to typically developing children. And this is relevant to performance. So the higher the static modularity, the more symptoms the children with ADHD experienced. And this is, as I talked about before, I'm interested in how state changes may impact both time-varying functional connectivity, brain network organization, and also behavior. What's I find interesting here is that this is a more, I would say, trait measure of functional connectivity that remains relatively consistent from day to day, and this is related to a relatively stable measure or trait measure of ADHD symptomatology. In terms of the network variability, I found that in ADHD that was related to response time variability on a continuous performance task. So just like in Aaron Kusi's paper, when functional connectivity varies more, when brain network organization varies more from moment to moment, you see more variability in performance, which is thought to relate to decreased attention. And I didn't include it here, but these differences are, for the most part, distributed across the entire brain. But the default mode network in particular seems to be driving a lot of these differences. So there are strong connectivity differences, the default mode network, for the typically developing and the ADHD children. And so this is in resting state. But what more can we learn from this? So one thing is we can start to look at this data in tasks, which I'm collecting some data now doing that, and we can ask during task performance, are periods of high default mode network related connectivity, are they related to high response time variability or other markers of inattention? And conversely, are periods of low default mode network related connectivity related to low response time variability, perhaps indicating greater attention? And in terms of clinical relevance, specifically for ADHD, are there more periods of high default mode network related connectivity? So in other words, are these patterns the same across all individuals? And the difference is the time in states or the transitions across states or what states individuals tend to be in more. And then more generally, do patterns like this relate to symptoms or to cognitive deficits that we see in different patient populations? So I just want to conclude by just summarizing. So it's known that dynamic neuronal signaling is critical for behavior. With time-varying functional connectivity tools, we can measure proxies for these neuronal dynamics and relate them to external measures of behavior and cognition. So I talked about some research today showing that time-varying functional connectivity is related to individual differences in cognition, and also that time-varying functional connectivity is related to variability in behavioral performance within an individual. So we see this both within individuals and also across individuals. And these external measures validate the utility of using time-varying functional connectivity measurements to understand brain dynamics. And finally, these tools can increase our understanding of the mechanisms of cognition in both typical and atypical populations and how they may differ from each other. And finally, I just want to thank my lab members who've been involved in this research, and also Stuart at Kennedy Krieger Institute and Mark at UC Berkeley, where I did my training, the data I talked about was collected in both of their labs. So thank you. Any questions?